Well, just as a joke, so Victoria invited me to share my summertime pictures, vacation pictures. So this is like a project started by an artist called Daniela Polis, called Cogito. So I invite you to check the website. It's brand new setup, Cogito in Space. And basically, it's kind of an extreme neuroscience project. It's more from the art side than the science side. But so here I'm programming, and this is a 30-meter dish for radiotelescopy, and we're like basically sending EEG into space. So, well, that's an extreme case of extreme neuroscience. But what brings me here today is not at all sending brainwaves in space for poetry, but more like social neuroscience, which is also a form of extreme neuroscience in a way. And I want to share with you some results about diversity in neural and behavioral dynamics. So as a start, I want to share like this small video from my PhD supervisor, Jacqueline Adel, who studied during 30 years imitation in infants. And I want you to put attention on the behavior of these two kids, how imitative and synchronous they are in their behavior. And even before like having language, they are like producing sounds and responding to each other. I love this video because it ends by a very humanistic move of sharing the puppy. Yeah, so, well, that's what motivates me in a way, to understand what makes us so social and how we construct our cognition through interaction with others from the beginning. And the problem and the paradox is that, as Pfeiffer and colleagues put it, is that there is the dark matter of neuroscience is social interaction. So if you look at the literature of neuroscience and neuroimaging, when you look at study with two individuals engaged in a dynamical interaction, there is basically not so much. And that was the start of my research project that covered many different methodology. In 25 minutes, I would not be able to cover all of that. So I'm going to focus on hyperscanning EEG. But I encourage you to check out our paradigm called the human dynamic lamp about human-machine interaction with bio-inspired models, and also some computational models that explain what I'm going to present in the experimental level. So hyperscanning, what is that? So if you check on PubMed the number of papers in hyperscanning, you detect that there is a small trend, kind of. And if you ask me why I started in 1965, it's because the first paper in hyperscanning actually comes from two ophthalmologists, and it was published in Science in 1965. Unfortunately, it was about extrasensory electroencephalographic induction between identical twins, which was categorized as parapsychology. And the only figure of this science paper was like electroencephalogram like that. So that maybe explained this kind of desert of publication after this paper, where like people were not really enthusiastic about this technique. But fortunately, in 2002, we have like a rebirth of hyperscanning, and the term was coined at that time because it was in fMRI by the team of Reed Montagu, and they expand the idea of behavioral fMRI synchronization with another fMRI, so you need two fMRIs, like costly a bit, but at the technical level it consists just at coordinating the acquisition of the two recording through a server. Not so much after, the team of Fabio Babiloni in Roma, and I cite the Rolling Stones for the EEG because that's good for behavior to have timing at the millisecond time scale. They start to look at interaction like a play card game. So it's called Trete, it's an Italian card game. And they checked how the dynamics of the brainwaves of the people in the same team were more similar than with the dynamics of the other team. And it was already interesting because they extract informationally this similarity in the gamma band, which is high frequency band, like around 40 hertz. But it was like kind of average across the whole interaction. And what brings back to my project was Jacqueline Nadal, so my co-supervisor, who pioneered in this kind of behavioral hyperscanning by using a dual video system for study mother-baby dynamics, and showing that at 3 months old, babies are able to differentiate a pre-recorded video of their mom from a live mom on the TV. And we kind of take the same idea and added 2 high-density electroencephalographic recording, so measuring the brain waves of the people while they are interacting by this dual video system, and using spontaneous imitation as a paradigm. And here you have the video. So that's how the people are seated with their arm rest on a table to prevent too much artifacts at the motor level. At the beginning, there is just a baseline with no view, no motion as a kind of control condition. Then there is this no view motion condition where they still have nothing on the screen, but they start moving. But since they are moving together, it's kind of another level of control. And then comes the spontaneous imitation case. And here you see the frame-by-frame analysis. As I understood in developmental psychology here, a lot of people are doing this kind of painful analysis. And here it's synchrony, here it's imitation, and here is the who is leading the interaction. And you can see that in the same way of the 2 kids we saw at the beginning, there is a kind of turn-taking occurring at the nonverbal level between these 2 people. And we ask them, the instruction are to move their arms continuously, not stopping, and imitate the other when they want. So, and to not do symbolic gesture. So it's purely like dance. Yeah. So, well, it took 11 months to analyze this frame-by-frame stuff. So I let you take a glimpse on that. I was doing a nightmare at the time about this software. And then we had to find, you know, maybe more than me, other people to do the same thing, to do the kappa coefficient and so on. So it's kind of, yeah. Anyway, so we have 25 minutes, so I will skip. So at the behavioral level, just already to match with the existing literature, we found that the subjects were primed to do that, but we were happy that more than half of the time they were in synchronous imitation. So doing the same movement at the morphological level, but also starting and ending the movement together. Then the second is synchrony without imitation, so starting and ending the movement without necessarily doing the same movement at the morphological level. Then imitation without synchrony, so doing the same movement morphologically, but not at the same moment. And finally, what we initially called no interaction, which was no synchrony and no imitation, was actually interesting because it's occur when the turn-taking occur. So like even in this case where we say it's not social case, it can have a social meaning as well. Like I'm bored, I stop imitating you, it's my turn now, that kind of thing. So how we met the bridge with the neural level, basically we take the EEG recording and for each electrode, so here we take the same electrode on the 2 heads, we can filter the brain electrical activity, extract what is called the phase, so how the signal is in the cycle, and we have a phase locking value, so it's a mathematical measure that quantify how much the 2 signals are in sync. But I won't go into much mathematics. You can already see qualitatively that those signals seems more in sync than there, but this is a more objective way of quantifying it. So if you do that with all the electrodes and across time you have the dynamics within one brain and across the two brain. And here you have like in Battleship all the electrodes, and so this subject 1 with subject 1 electrodes, this subject 2 by subject 2, and those are like the interbrain part. And here I plot the histogram for the intra and interbrain locking. So how the signals within brain are synced and how they are between brains. And so you see the antero-posterior synchronization that are well known at the intra-brain level, but you have this burst of similarity between the two brains occurring during the interaction. So same thing, I referred to the paper, but there are a lot of methodological issue with that to control everything. It took me like 3 years to publish this result because it was kind of unusual type of analysis, you would bet. And we demonstrate that with 2 different methods to be sure that what we were measuring was what we were measuring. But so like the basic summary is that indeed there are synchronization that occur between brains, and they occur in different frequency bands. So as you may know, in electroencephalography you have different rhythms in the brain. These different rhythms have been correlated with different behavioral characteristics. We are far from understanding their exact meaning. We have to, for instance, the alpha mu was discovered by Ansberger in 1929 has more than 50,000 papers on it, and still we don't know really what it is about. There are models, but it's not set. So the alpha mu was the strongest, actually, synchronization between the two brains, but interestingly, the beta and gamma, who are more associated with attentional effects, were also involved and more asymmetrical, kind of retranscribing the social role in the interaction. But interestingly, so that was the most funky result in a way, but at the intra-individual level, we took also the advantage of a hyperscanning experiment to actually investigate the different level of agency and correlates of how we attribute agency to others and to ourselves during social interaction, which was not really the case because most of the study were made from an isolated individual point of view in a passive video watching. So here we have real social interaction, and what we found is that beyond the difference between social perception and social interaction, which is like the basic and most important take-home message, is that definitely we have to study social interaction because it's categorically at the behavioral and the neural level different from social perception. There is even more fine-grained neural correlates, especially depending on the role you take in the interaction and depending on the context of the interaction. So in the same way, here you are experiencing a monologue, and maybe it's boring for you because it's unidirectional. It's called like induced social interaction. There's a clear mark between imitator and model in the theta band. In the spontaneous level, when like we are at the pub drinking a beer, and more like improvising the discussion in a very spontaneous way, there is different patterns at the neural level. And especially one pattern, which is like if you play the mistakes, it's in the fMRI results done by Guillaume and colleagues at the Pitié-Salpêtrière in parallel to our work, that show that there were differences in the reward system. Especially there were a reward in spontaneously interacting with others that were not present in the induced case. And so since we have a lot of people in education, science, I have seen, I have wrote 2 reviews with a very interesting collaboration with Jan Clark in Nagoya in Japan about what these neuroscience results can tell us about education from a more evidence-based perspective. And especially what I show you about social interaction and reward has a lot to do with peer interaction and how it plays a role in the rewarding of learning with others. And so I had the chance to discover now the literature of education science like Bandura's and Vygotsky. It was very interesting to put these two kind of literature aside. But I'm more in the medicine and biomedical research at the Institut Pasteur. And so now I'm starting a platform called Soneta for social neuroscience therapeutic approach in autism, and working in the Hôpital Robert Debré in the northeast of Paris, who was already pioneered in the genetics of autism with Thomas Bourgeron and Richard Delorme, and in neuroanatomy of autism with Roberto Toro. And so I have created a new platform for social interactive investigation of autism through hyperscanning EEG, but also this new paradigm that I mentioned, but I won't have time to discuss, which is the human dynamic clamp. And so what we do is actually to have a mixed and controlled dyad with similar experimental design for the hyperscanning scale. I have done some automated behavioral analysis because I didn't want to have nightmare anymore with this frame-by-frame coding. So fortunately, machine learning is blooming, so we can use that to do this kind of analysis. And we update also our way of plotting the results. But what I will show you, because I wanted to share about neurodiversity, the first results were like kind of surprising because it was not at all what was interested in the first place. We were trying to replicate just results about the mu suppression, which is a phenomenon that occur when you do an action and when you observe an action, there is a change in the brain rhythm that is similar. So like it kind of connects at the literature level with what people have called the mirror neuron. So these neurons that spikes when you do an action and when you see someone else. And the broken mirror theory was proposed by Ramachandran and Lindsay Oberman, state that basically in autism there is a problem with these mirror neurons. And many people replicate their results of difference of mu suppression during execution and observation in autism. But as Einstein famously saying, insanity is doing the same thing again and again and expecting different results. So they were replicating the same methodology, observing the same results, but actually it's not that clear. And especially we replicate their results as well. But it's not that simple, especially they take, we were talking about dynamics, and all this literature take C3, C4, so 2 electrodes in the large alpha frequency band. So if you take all the electrodes on the scalp in high density, like here, 64 electrodes, and you take all the frequency, all the rhythms, and you observe what's going on, indeed, in the control group, you observe this effect of suppression, so like reduction of alpha mu rhythm. But in the case of patient with autism, we didn't observe a reduction of the suppression as the average would have tell, but actually 2 different trends, like a reduction that is normal, like in the control in the lower band of alpha, and an increase in the upper band. And so if you summarize the result by taking the lower and the higher band, and this is the average, so indeed you find the original difference published by many people. But if you look at the difference in the lower and upper band, there's no difference in the lower band, but a big difference in the upper band. And it gets more interesting when you look at the SCAP results, because it's not at all at the sensory model level. So the C3, C4 electrodes are here and here. Actually, what we observe is a normal there, and you have the increase in the frontal region and the decrease in the occipital region, which is like not at all what people were reporting by taking only C3, C4. So as Claude Bernard would have said, by destroying the biological character of phenomena, the use of average in physiology and medicine usually give only apparent accuracy to the results. And by taking these averages and kind of negating the dynamics, we are kind of hiding what's going on. So what happened at the interactional dynamics? So we also analyzed the imitation patterns between the mixed dyad with neurotypical and autistic patients. And we found, surprisingly, that imitation was not impaired, but more increased in the patients. And what was changed was synchrony. And if we look at the repartition of the role during the mixed and neurotypical control dyads, there is a strong imbalance with a strong imitation perspective from the Asperger participants in the mixed dyad. But so if you notice, there is a problem here. No, you don't see? So I say that averages are bad. So don't use bar plot. Friends, don't let friends make bar plots. So I recommend you to use violin plots. It's better to check about the diversity. And so there is like even a Kickstarter. You can have a T-shirt about bar plot. So if you use violin plot, you have a better picture of what's going on. And actually in the autism, that's also something that have hidden a lot of what's going on, because people were taking the autism as a construct, like a social construct as a case. Whereas like if you check the behavior of, for instance, the mixed dyad, we have a huge heterogeneity of behavior. And so that's actually what characterizes maybe the most what I see in autism is the huge heterogeneity of behavior. And so the lab I joined at the Institut Pasteur pioneered in the genetics of autism, and they show that behind this strong heterogeneity of autism that is already seen in the comorbidity, because you have a lot of different comorbidity from there's big difference between a patient with Asperger's syndrome and a patient with intellectual disability, with strong epilepsy and so on. So we have to dig into this heterogeneity, understand how diverse is the system. And at the genetic level, there are more than 200 genes that have been associated with autism now, which show that the biological underpinning are also heterogeneous. So what we are working now is to combine systems biology and social neuroscience to do patient stratification. So basically finding subgroups of patients within a large cohort. So we have like collaboration, especially in UK with the European Autism Project EU AIMS. And with large number of participants, we can dig into this heterogeneity, and instead of having averages, we can go back to the individuals, characterize them at the molecular level and also at the phenotypic and endophenotypical level, and cluster them to find subgroups that are more homogeneous. And this is basically with the same ideas we have seen in the earlier talks, to have a more personalized medicine. And so instead of having, the DSM-5 say you have this, so you are autistic, the idea here is to go back to the individuals and find out what are the best behavioral techniques or pharmacological intervention that we can have based on their subtypes. So I'm actually super fast. I thought I was scared to overdo the talk. So I have already had the take-home message. So we have more time for interaction after. So the take-home message, as you saw, is social interaction is different from social perception. That is like the major take-home message. Okay. The second take-home message is that it's way more complicated than what people would say. At the social level, we are intertwined between so much layer of factors. And especially when we are dealing with hyperscanning and those measures of similarity of brain waves, we are measuring similarity that are coming from different level of organization. So there is like, first, if indeed the neurobiological similarity, so we are obviously more similar than we are with like a snail, for instance. So like our biological similarity help us to share similarity at the behavioral and neural level. At the behavioral level, because of course we are doing behavior together, so I used kind of extreme case, which is imitation, where like imitation is about similarity. But there are like a huge amount of literature that is about joint action and how complementary action are on our daily basis a big deal. So at the behavioral level, we have also similarity. And so I didn't have time too much to talk about that, but for instance, there is a lot of work, for instance, by Ivana Konvalinka in Hyperscanning, which is more about how the neural patterns are also complementary, but mimicking each other in an asymmetric way. So in the same way I show leader-follower, it can extend to complementary role in a joint action schema. And finally, there is also similarity at the cultural level. So I was really enthusiastic by the talk, the first talk, about having a cross-cultural approach. And I don't know if you're familiar with this, the weirdest people in the world paper about anthropologists questioning how normative we are about studying white male, right-handed from industrialized country. But this kind of a problem also in the literature because we have a very biased sample. And beyond this similarity that we are measuring, that of course is interacting because neurobiology affects the behavior and behavior affects cultural, there's also communication, so the sharing of information between the two people. And we saw that even within this big box, there is like unidirectional kind of interaction and exchange of communication and reciprocal. And from my perspective, the key challenge, and that's why it's important to have interdisciplinary dialogue, for instance, with educational science, is that the way we communicate actually affects the culture we are building, and the way we are biologically grounded also affect our capability of communicating, like in autism. So yeah, that's a big challenge ahead. And the last message I wanted to share, it's about this stratification and how we are moving right now from this case control way of thinking. And I would say that even in more general case, even without talking about psychiatry, the way we are dealing with contrast in neuroimaging is also changing. There is more and more people doing parametric designs. And instead of having this dichotomic vision, now we can have either stratification or more like continuum spectrum or even mixed case. So that's a very interesting biological psychiatry paper about these normative models. So those are like all the people I had the immense chance to collaborate with and the fundings. And thank you for your attention. So if you have questions.
Thank you so much, Guillaume, for that absolute tour de force, and he is indeed breaking barriers on our behalf. Your first paper took 3 years to publish. I really hope that our synchrony paper will not take half as long. No. Right, so I'm sure there must be questions here, so can I open up the field for some questions? It's just
I can shout. I can shout and record. So just a relatively wide question about the relationship between the genes and the EEG in this interaction of
scales,
two people.
Yes.
You have any thoughts or anything about projects that are sort of pushing this idea of interaction? It's in the present, and you're looking at brainwaves and behavioral interaction. What about all the other things that are slightly more static or trait-like? Do we have a model how to treat them? Because the way I'm thinking about it is you measure in a chunk of moment in the present. Two people interacting in a particular way, either following each other, not following each other, imitating, etc. And then there's a amazing amount of information around that, which is what's the trait of the person and the state of the person there. That could be genetic, could be physiological, etc. Have you given any thought about how to include these
yes.
static aspects to the dynamical aspects?
So that's part of the slide that I have to skip.
This was we already
So
talked before. You know.
the way I
ask
see
you about
it is
that
to
one.
use computational modeling to make the bridge between the different scales. So for now, what I worked on is how to bridge the gap between structure and dynamics, especially how the anatomical connections are constraining the EEG patterns. And the idea is that I took what's called the human connectome, so through the fiber bundles extracted by these diffusion tensor imaging techniques that do these beautiful pictures. And at last you can build up kind of backbone at the anatomical level, and by adding oscillators for each nodes, you can simulate oscillatory activity on the network and check how the anatomy affect the dynamics. And the dynamics at the intra-individual level, but also at the inter-individual level. How untrainable we are to each other by sharing the same anatomy. And the idea is that when you share the same anatomy, you would tend to have more ability to coordinate, for instance, at the sensory model level. And what characterizes, as I say, autism is especially this strong heterogeneity. So that's a very interesting paper by Hami and colleague in Nature Neuroscience, where they show that in the same way, averages of autistic patients tends to affect the way we saw. So here you have controls, and this is the average on control groups. And this is like ASD1, ASD2, and ASD group. And then you say, oh, it's weaker activated. But no, it's not weaker activated. It's just that the patterns are so different that when you average anatomically, you get a weaker response. But so like this idiosyncratic brain, as they call it, could explain also maybe this difficulty of propensity to coordinate with others at the sensory model level. So that's one part of the perspective I'm trying to dig in. The other part is how highly genetically constrained our biological rhythm. There are a lot of genes implicated in autism that are related to synaptic cleft. And my boss in Pasteur discovered these first genes that are synaptic scaffolding proteins. And these genes obviously strongly affect the way neurons connect to each other, and also then how brain rhythms are produced. And so if your neuronal machinery is out of tune at the biological level, it makes you also prone to have difficulty to synchronize and coordinate with others. So that's another way of bridging the gap with genetics.
One more question? Oh, there's two. I think we're going to have to go. Yo, thank you for being a gentleman.
Do we have any ways of combining measurements such as from EEG with other techniques such as real-world body movement? Do we have any research on that?
So it's good. I have all this slide that I skipped. Now I can. So body movement is kind of tricky, but what I have in mind is already to dig into eye tracking. So because like EEG is really easily artifacted, contaminated, but gaze and is constitutive also to our joint attention and our social world in the early life. And there are like, I have collaborators like Leonard Schilbar in München, who are working on this dual eye tracking system. And now I'm setting up in Paris this system combined with dual EEG. So to have like joint attention paradigms. I know that in San Diego, there are people doing with this called MOBI platform, like full motion capture plus EEG and so on, but it's highly challenging. I think for now we are more at the methodological level to remove the artifact and so on. Yeah. But now eye tracking is, I think, production ready for, yeah.
Fantastic.
They are doing that kind of research with mirrors though, so they have adults walking around squares in London doing locomotion tasks and thinking about spatial awareness and traffic handling and things like this. So they've got caps, because that's less susceptible to artifacts, so they can do sort of actions more easily
Final round of applause for Guillaume, please.